my goal today is really not to, is to present a few tools that we've been working on in various collaborations for projection analysis. And I'm going to quickly define what I mean by that and how we're trying to use this, at least in my group, to study the mechanisms and gene regulatory networks that are associated with fate commitment in neural development predominantly, and also changes in cell state over neurodegeneration. So I should also outline specifically that given the brevity of the time, or the short amount of time that I have to describe these tools for you today, I'm going to focus less on the results and more just kind of give you an overview of what these tools are and how we've used them in the past and how we can expect to use them in the future. But I want to just start today, if possible, by reminding you that I think almost everyone here is familiar with dimensionality reduction as a tool for modern biology. And I think, given that modern biology is moving towards significantly higher dimensional data analysis, I would imagine that almost everyone here is at least familiar with the concept of dimensionality reduction to reduce understanding and to simplify the data by collapsing it into a reduced set of latent spaces. And most people here have used this in one form or another, either by using principal components analysis, singular value decomposition, TSNE and UMAP are predominant mechanisms for dimensionality reduction these days. And also some of the more recently developed tools that are centered around neural networks like SCVI for single cell analysis, in principle do the same thing, as they take these large matrices of genes versus samples. And in the cases that I'm most familiar with, we're using single cell RNA sequencing. So we have this high dimensional data set that we want to actually reduce into a meaningful set of latent spaces. Most of these methods accomplish the same goal, but have different optimization functions. But under the hood, each of them is identifying a series of what we call hidden dimensions or latent spaces, latent representations of different features that summarize biological properties and or technical properties such as noise of the data. And what I'm going to share with you today are 3 tools that our lab has developed that actually help you go dig a little deeper into these latent spaces. The idea is not to just kind of superficially use them for classification or clustering, but to really do a deep dive into some of the biological value that we can get from individual latent spaces. And most of this has been, there's been an explosion of this study since we've gotten to single cell resolution of cellular measurements. And the idea that underlies all of this is that these individual latent spaces reveal what we like to call axes of cellular identity or cellular features, and that different properties of individual cells will allow them to be positioned along these independent axes when we do this dimensionality reduction. So when we reduce a large data set of genes versus cells, we can identify individual spaces that might describe specific features such as cell type, spatial position, their position within a state like cell cycle or a differentiation signal, or some other continuous phenotypes such as oxygen sensing or a metabolic activity. And the way that we like to, in my lab and through our collaborations, analyze a lot of these data is by employing these latent spaces individually, either in a supervised way or unsupervised way. When we learn these latent spaces, we gain insights into specific biological features and properties that we can then transfer across different experiment types using some of the tools that we've developed. So a brief overview, so the tools that I'm going to describe for you today can actually use as input any latent space that you describe. So if in your data you're using principal component analysis or independent component analysis, or even a variational autoencoder, the hidden spaces that you get, the latent dimensions that you get from this analysis can be used for the types of tools that we have. We like to focus on non-negative matrix factorization, and this is just a brief overview of what that actually does. Is it takes a matrix of gene expression values by cells and factorizes it into 2 different matrices, the product of which is a reasonable approximation of the original matrix. And I don't have a pointer here, but I'll just use my voice then. The 2 matrices, in this case, A defines basically the gene weights in a specific latent feature. How much does each gene actually contribute to the signal or identity of what we like to call a pattern or latent space? And then matrix B actually corresponds to how those latent representations are actually used across the original input cells, okay? So the benefits of non-negative matrix factorization here are that the solutions to this, to solving this, are actually constrained to be non-negative values, which is consistent with what you would see for normal gene expression estimates. We don't actually have negative gene expression. And in collaboration with Alana Ferdig, we have a postdoc in our lab who helped develop this single cell coordinate gene activity in pattern sets, or SC-CoGAPs, is a tool that basically is a Bayesian non-negative matrix factorization that also includes an adaptive atomic prior to deal with the sparsity that is inherent in single cell RNA sequencing. So this is the only slide I have on how we actually learn them, but the idea here is that we're interested in the columns of A, and we're specifically interested in how the columns of A represent individual features of cells, like cell type, metabolic state, the specific use of a gene regulatory network in a developmental context. And this is the tool that we prefer to use at this point, and we've developed and is available, but any pattern discovery or latent space discovery tool is applicable for what I'm going to show you today. So here's the overall concept. And this slide introduces the three tools that I'm going to talk about in a little bit more detail. But the idea is that doing the dimensionality reduction and then a UMAP gives you, meaningful interpretations of things like groups of cells that are related, that are independent, that are related to each other and maybe independent from each other in terms of the groups. But these patterns themselves, or these latent spaces defined as columns here, the A from the previous slide, these are actually, the analogy here is that they're lenses into specific features and processes, such as I said, metabolic state, cell type, or even disease conditions. So you can actually take these patterns and project new data into the space defined by these latent spaces and reveal the use of those patterns in new data sets. And conveniently, the way that we've done this is that this happens almost instantaneously. It's a multiple linear regression in our original implementation of this in Project R. Excuse me. So individual patterns can actually be evaluated in a matter of seconds once you have a new data set. And you can really see whether or not cells or specific samples that you have, and it's not just restricted to single cell analysis, whether or not these cells or conditions are using a particular biological feature or property. And you can also use this to segregate, for example, disease versus normal samples in a very rapid screening approach. So as I mentioned, SC-CoGAPs learns these kind of biologically meaningful axes from single cell data. And we've developed three different projection tools, the first of which is Project R, and the links will pop up periodically throughout the talk. Project R is the R version of this, and we have a new tool that's coming out shortly, Scproject, that builds off the Scampi platform for single cell analysis in Python. And then we have a specific flavor of this called Tricycle that actually is designed to look specifically at cell cycle and in a projection analysis. The first example I want to give you is a paper that we published about a year and a half ago on retinal development. And as I said, my group is interested in the development and fate specification of individual neurons, and also how the cell cycle kind of contributes to competency of the progenitors in specifying different fates. And this is just a model of the temporal organization of how different types of cells are produced in the developing retina. And in this study, we actually did a single cell analysis on about 120,000 individual cells from various stages of the developing retina. And those stages are kind of color-coded here, from red being the earliest and pink being the latest. And when we condense this data and analyze this data, we actually get this manifold that very neatly describes a whole bunch of biological processes, including progenitor maintenance and differentiation along this kind of longitudinal axis on the bottom, exit from the cell cycle, and then of course specification into different branches of major lineages and different cell types. So as part of the study, we actually did a deconvolution here with SC-CoGAPs and identified 80 different patterns of coordinated gene expression or latent spaces from these. And many of these patterns correlate with different ages for the samples or different cell types. And there's some other features that actually are shared across different cell types and time points as well. But the important part here is that this deconvolution into the latent spaces reflects distinct features of individual cells and populations, regardless of the sample in which they were actually derived from. So here I'm showing you one pattern up the top that defines cells that are in a G1 to S transition, and another one on the bottom here that actually defines cells based on the sex of the embryo that was used. As I mentioned, my lab is interested specifically in fate commitment and developmental specification of cell types. So in this context, we were specifically interested in this late neurogenic and late progenitor identities that we identified as independent latent spaces. And the value here is that we can actually go and look at the genes that have high weights in these latent spaces. And we initially identified a group of 3 related transcription factors, the NFI family A, B, and X, that were specifically enriched in this particular latent space associated with late RPCs and commitment to glia, which is a late-born phenotype in the neuron. If we look at the gene expression profiles of these individual genes, we can actually see that in many cases, they're actually used in different lineages to help specify different cell types here. But the thing that they have in common is they're actually all induced in this late retinal progenitor state, which was something that we hadn't observed before. So briefly overexpressing these 3 transcription factors together, we found that in the developing retina, we actually found that it promotes the identity of late-born neuronal types like bipolar cells or glial types like Mueller glia. And this comes at the expense of early-born cells like amacrine cells or retinal ganglion cells. And furthermore, if we knock out this population, we can actually see that we get increased proliferation in the progenitors and a reduction in the late-born late-born cell type identities within the neuron. They actually fail to produce Mueller glia, and they actually never leave the cell cycle. At least 30 days postnatally, these progenitors are still dividing within the developing retina. So this is a way that we actually learned these genes and their role in quiescence and fate specification in the retina by exploring a specific latent space that had usage at a developmental time point that we were particularly interested in. So this is to just highlight how individual spaces can contain the information that is relevant for specific biological processes that you're interested. We also have 80 of these features that we need to explore, and we wanted to actually see what is the utility of these, or what is the value of these individual latent spaces in other cellular contexts. So we started to think about how we might actually use these individual latent spaces, and we developed a tool called Project R that basically takes a new target data set and performs a change of basis using a multiple linear regression to actually embed those new cells into the latent spaces defined by the source data set. And in this case, this is just one of those patterns, which is a rod-specific pattern or latent space that you can see in our original data. And this is just another UMAP here on the left of our retinal data. And you can see that this pattern is highly used in the rod population. And if we project another data set, in this case, this is now adult retina, not a developing retina, we can see that the use of this rod-specific pattern is actually very specific for the rods, with a little bit of exception in terms of background expression in other cell types. So we can actually quantify this. We can actually see just how good each of these patterns are at recovering known annotations. And we can see, for example, here that the rod-specific pattern has very good predictive power for rods in this new target data set, versus another pattern, which is specific, again, that cell cycle specific pattern, which you can see basically has no use in this adult data set because these are all post-mitotic quiescent cells. And in this way, we can actually use each of these patterns that we've learned to predict and de novo annotate cell types, again, in a very rapid way between data sets. And if you're interested in this particular approach, we go into a little bit more depth into the paper, and I'm happy to share more detail with you about that later. But it goes just beyond retina to retina comparisons as well. We can take those same 80 patterns that we learn in the retina, and through the same multiple linear regression analysis with Project R, we can see that these patterns are actually used in different cell types. And here, this is the original tabula muris data set, which is a multi-tissue single cell analysis, where we can see that these individual patterns project to different cell types in different tissues, meaning that these processes that are identified and learned are identified and described by the latent space are actually being reused in different contexts, not only developmentally, but here in adult tissues as well. And importantly, this is not just across tissues as well, this is also across species. We can do the same projection in a developing human retina and see in the middle here, we have single cell analysis from the developing human retina. And we can see that these processes are, again, shared, the two that I'm showing you here in the developing human retina, but we can also do this for bulk RNA sequencing. So if we look on the right at columns corresponding to bulk RNA sequencing samples at different developmental stages in the human retina, we can see that there's a very reasonable and nice progression from earlier used patterns such as cell cycle neuroepithelium commitment, again in mouse, and that these are used and conserved in the human early development, and this progresses to more of a cell type identity set of patterns down at the bottom. So in this way, we can go between human and bulk as well. And one thing that's potentially relevant here for the group is that it also works across experiment types. So we can do this, you're looking here at ATAC-seq peaks around transcription start sites for different, in the mouse retina at different developmental time points as well. And there's really no coordinated regulation of this signal. And in fact, there's some batch effects that you can tease out here in the raw data. But if we can actually project these data into the patterns defined by gene expression here and can actually get differential accessibility types over transcription start sites for different patterns, including things like the pattern 31, which is the cell cycle pattern, or the second panel here, which is pattern 21, a rod specific pattern, which actually doesn't turn on at these developmental time points. So you can see that that's flat. So an example of how you can go across things like gene expression and ATAC-seq to make inferences based on the definition of these patterns. Very quickly, I'm going to mention that we have another tool that extends this. This is Scproject. This is we're going to release the preprint for this in a week or so. So if you're interested in using this in a Python work environment, you can also use the Scproject instead of ProjectR. And this uses a different elastic net regularized regression, which has additional benefits that I don't have time to go into here. But we've also identified we've also developed a projection driver statistic that identifies pattern-specific differences in gene expression between conditions. And just to give you an example here of how we've applied this, and unfortunately I don't have time to go into the details, is we apply this pattern driver statistic to human developing mouse retina and developing human retina. And if we look at the gene expression differences between photoreceptors and all of the cells, we can use this, and also through the lens of a particular pattern here, 75, which is a photoreceptor specific pattern, we can actually weight the fold change difference based on the gene usage in the pattern and prioritize and basically rank genes that are different between these two species. And I'm just giving you a few examples of genes that we found here that are different between mouse and human photoreceptors. And we validated one of these dopachrome tautomerase because it was surprising to us that we actually saw specific expression of this gene in humans, in human cones. And this was actually only thought to be in the RPE, but we've actually further validated that this gene, which is a precursor, which is an enzyme involved in melanin synthesis, that this actually exists in humans. And here on the right, we can actually see in the second row that this is actually human specific. If we extend this analysis to include macaque as well, we don't see evidence that this is differential between human and macaque. We don't see that this is let me rephrase that, we see that this is a human specific feature. So basically using this projection drivers tool, we can actually go in and look at differences in pattern usage between species or conditions. The last thing I want to tell you about here, and again, I apologize for going through these so quickly, is a tool that we just released on bioRxiv a couple of weeks ago called Tricycle, that is specifically focused on the cell cycle and doing a projection analysis in a well-defined reference space that allows us to make continuous estimates of cell cycle position, not just binning things into G1, G1S, G2, or M, but actually getting a continuous measurement as indicated by this, the legend here of movement through and progression through the cell cycle. It's very compares very favorable to experimental measures, and importantly, is highly scalable. We did this projection of a human fetal tissue atlas that you're seeing here in about 5 minutes using this regression tool, Tricycle. But basically, this stemmed from an observation that when we did principal component analysis on data sets where cell cycle is the greatest source of variation, we often see an ellipsoid shape. And this ellipsoid shape is seen in different data sets, in this case, on the left, mouse neurospheres, and on the right, mouse hippocampal neuroprogenitor cells. And these principal components seem to be breaking down very similarly across these proliferative data sets. So we actually did a deep dive into some of the math behind this, and it turns out that when you do PCA on periodic functions, which is what you usually get from most cell cycle regulated genes, and periodic functions with a single peak, meaning genes that only peak once during the cell cycle here, the result is an ellipsoid in PCA because the variance covariance operator is determined by a two-dimensional matrix. And importantly, this is not the case when you look at data sets that are not dominated by cell cycle in the first principal component, as you would expect. So looking at this pancreas data set, developing pancreas data set of principal component analysis of these data, you can see that the differentiation state is the predominant component, and we don't actually recover cell cycle from the PCs. However, if we just subset to the progenitor cell, the ductal cell here, we actually do recover that ellipsoid. So our solution to this was to establish a reference cell cycle latent space. And we used our mouse neurosphere data here to define that space. And then we can actually use the angle of rotation around the origin as to infer cell cycle position in this case. And so we can project all of our new target data into this reference latent space and robustly estimate cell cycle position as the radial position around the origin. And importantly, projecting into a common meaningful latent space allows us to actually make correct comparisons between different conditions. So here I'm just showing you very briefly the result of this on some of these data sets that include not just proliferating cells, but also differentiating and postmitotic cells as well. And in each case, if the data set includes cells that are cycling, we get an ellipsoid shape, as you see from the top row. And if we look at the expression of specific genes that are known to be regulated within the cell cycle, they're at very similar positions along our theta estimate here. And down at the bottom, we're showing you a UMAP of the top few 10 or 20 principal components from each of these data sets, colored by our now cell cycle annotation, and you can actually see very cleanly the progression. And importantly, it is continuous and not discrete, but this method is also very robust to sequencing depth. If we downsample the libraries, we can still get very robust and accurate and consistent estimates of cell cycle position. And importantly, and this is a source of future direction for me, is that the shape of the ellipsoid is actually well correlated with cell cycle dynamics over retinal development. And you can see here again that developing mouse retinal data set that I showed you before, embedded into this reference cell cycle shape, we see changes over developmental time in terms of the size of the ellipsoid, which potentially correlates with changes in overall variance within the data set, and in the percentage of cells that are in that ellipsoid versus those by the end of the P14 that are all postmitotic and relegated to this G1, G0 puncta here. So this is the quick introduction that I had to these tools. I'm very happy to discuss either in questions or offline how you might be able to use these tools, what they can do for you. Here's just a smattering of the kinds of applications that we've used and we're developing for these projection tools. We're very interested these days in looking at how we can project between gene expression or even ATAC measurements of genes and spatial transcriptomics. This is a Drosophila example, but we've been doing this in the cortex as well with Visium data. And all these other applications, including training classifiers or looking at disease-specific latent spaces and how they're used in larger catalogs of bulk data. So with that being said, I know the time crunch here is short, so just thank you to everyone from my group who actually participated in this and helped develop some of these tools, and of course our funding agencies as well.